Then let's move on to what you're going to do today. Well, I mean, you're going to be doing some culturing, but let's talk about the bacterial population counts, aka the viable plate count. And this diagram kind of is an overview of everything you're going to do. Now, I'm not sure why they're showing the original culture in what I would call a moonshine jug in the picture, but that's the way the artist, maybe he was thinking about moonshine when he drew that. I don't know. But there are what's the purpose of the bacterial population counts? What's the underlying purpose there? Who can tell me? What are you trying to do? Or what information are you trying to obtain from this? What? To count. What, Count Dracula? Oh. Right, the what do we call that? The OCD. What is OCD? I mean, I might be a little OCD or a lot OCD maybe, but in this context, what does OCD mean or stand for? Original culture density. Or you could even say original cell density. It really, in this case, means the same thing, OCD. And we calculate OCD in CFUs per mil. Now, there are instances in a clinical microbiology laboratory when knowing what the concentration of the culture is important. Knowing the concentration of the culture is important. And I'll give you an example. If you get a patient sample from a patient that has a bacterial disease, and you isolate the bacterial pathogen, create a pure culture of that pathogen, what kind of information do you think is going to be important for you to determine and be able to report back to the attending physician? Especially if this patient has severe disease.
CD.
I'm going to want to know what it is, but in addition to that,
what the count is.
That's not going to not in this case. And knowing what the concentration of bacteria in a culture that you produced is not going to help your patient. What additional information do you think would potentially be life-saving that you are going to provide to the physician if it's a bacterial disease? Think about this critically. Identity of the pathogen, extremely important. Beyond that, what do you think is most likely the next most important thing, perhaps even more important than the identity of the bacteria?
Treatment.
What?
Treatment.
Treatment, what do you mean? What do you mean? Say it again. Right. What antibiotics are going to work? What antibiotics is this species of bacteria susceptible to? Right? If you have a patient that is dying and you give them an antibiotic that is ineffective, you're not helping them. So one of the most important things that we need to answer in these cases are what antibiotics can be used effectively to treat this bacterial disease. So the testing process for that happens in the laboratory. By the way, you're going to be doing this in a future, you're going to be performing these tests in a future lab. But in order for them to work correctly and to be able to evaluate them correctly, the concentration of bacteria that you use for the test needs to be standardized. You need to know what the concentration is. You need to adjust the concentration to a standard in order for the test to be meaningful. Okay? So there's a method behind the madness of doing these types of things. We need to know this information. Okay? And so when we calculate the concentration of bacteria in a culture, we're calculating OCD and CFUs per mil. Now somebody remind me, what does CFU stand for? Colony forming unit. So what is a colony forming unit? one bacteria, that's almost correct, but you need to add something. Viable. It's one viable bacterial cell. Not a dead one, right? And viable most of the time just means alive, capable of reproducing. So one viable cell. Because if you put one viable cell on the surface of a plate, nutrient agar plate, and incubate it, that one cell is going to create how many colonies?
One. No.
What? One. Right. Just one. You guys are very tentative today. I'm not trying to trick you. I promise. Right. One. That's why it's a colony forming unit. If it's a dead cell, it's not going to form a colony. Right? So, CFUs per mil. Now, in the diagram here, oh, well, by the way, before I get to the diagram, this could be very, very simple, right? Colony forming units per mil. No problem. Let's take a nutrient agar plate, one of these TSA plates right here. And let's say this is one of your cultures. Let's say this is the culture right here that you want to determine the OCD of. You're going to do it for you're going to do this with the E. coli cultures that you inoculated on Tuesday that are now in the incubator. Those are the cultures you're going to determine the OCD of. Fine. Take a mil, 1 mil from your culture, spread it around on the surface of the plate. Not a 4-quick pattern. You just want to spread the culture around just evenly over the entire surface of the plate. 1 mil. Incubate it, and you take it out of the incubator on Tuesday, and there's 50 colonies on there. What's your OCD?
50.
You put 1 mil on here. Now you count 50 colonies after incubation. What's your OCD? You guys are thinking too hard. 50. 50. 1 mil went on there. If you get 50 colonies from that 1 mil, there you go. 50 CFUs per mil is the answer. How many cells per mil, though, do you think are actually in the culture like this?
Over 2,000, over a million.
Group 6, what do you guys think? How many cells per mil are in a culture like this?
1 mil?
One.
That's over a million, probably.
Okay, a million? That seems like a lot. Group 5, what do you guys think? Don't everybody answer at once. I want the rest of the groups to be ready with an answer. Group 5, go. 1,000. What, like 10,000? Pick a number.
Sure, 10,000.
Okay. What? 500. 500, okay. No, you're going to count them. How many? This is theoretically. Lucy,
That's what you want.
100? What? 10,000? Okay, what about you guys?
That's the ideal count you want.
No. There's an actual... That's a big range. No. How many? A million? 1,000? A billion? What? 200? 2 million. Oh. You guys are all over the place. Okay. Well, you're gonna find out, right? Okay, listen. You're gonna find out because you're going to perform this experiment and you are going to count them and calculate. Here's the problem with the scenario I just described. The number of bacteria per mil in those cultures is way too high. If it were actually 50, what I just described could work. The problem is they're so high that the size of the plate would have to be like, the size of LA County. And we would have to somehow magically be able to spread out 1 mil over a plate the size of LA County and then incubate, right? Can't do that. So the solution to that problem is dilution. You guys like that? The solution is dilution. The solution to that problem is dilution. So what we have to first do before we inoculate those plates is dilute the amount of bacteria way down from the original culture. So then we take a very diluted culture and take some volume from that, and then inoculate the plate with it and incubate it. So what you can see described or depicted in this picture here, is first it's a two-step process really, right? First you have to do the serial dilution. So you can see that there's that original moonshine jug culture, and then there are 5 dilution tubes. And then after you complete the serial dilution, you're then going to take an aliquot, a small volume from each of the dilution tubes, and use that to inoculate a plate, which then you'll incubate, and then after incubation, you will evaluate the plate and count the colonies on some of the plates. And if you do that correctly, your plates, you're going to end up with 4 plates on Tuesday. You're going to inoculate 4 plates today. If you do it correctly, your plates are going to look something like this. You can see, I should have labeled them, but it's from left to right, plates A, B, C, and D. Those are going to be your 4 plates A, B, C, and D. And you can see plate A, does that plate have the highest or lowest concentration of bacteria? Highest, right? That's got the most bacteria. And then from left to right, the concentration goes down. What you're trying to accomplish is you want to end up with one of your plates in a particular range of colonies. What's that range? 30 to 300. You need a plate that has colonies in that range. 30 is okay, 29 and lower is not. 300 is okay, 301 or higher is not, and anything in between is okay, right? So on the plate on the left, we wouldn't even try to count that plate. We would just call it what? Too numerous to count, right? TNTC, too numerous to count. The plate B, we would start to count it. How many colonies do you think are on plate B?
300.
300? How many colonies do you think are on plate C?
50.
50? What do you guys think? What, 70? 80? There's close to 200 colonies on plate C. I mean, I've counted a lot of plates, so I can kind of eyeball it and usually get pert near. That's a technical term for in the ballpark, pert near. So, and that would be the plate we would use. The plate B has way more than 300. We'd count it when you get to 301, just stop counting and go, oh, that's too numerous to count. I mean, you can count it, but why bother? We don't, right? So you're going to count these plates, you're going to evaluate them and count them, and then from there you'll be able to calculate the OCD. So now let's go back and talk about how we do that. Okay, so this diagram shows a serial dilution and then a plating, and then the results of that, the results after incubation. So I'm just going to put this as the original culture here, and I'm going to draw 5 dilution tubes up here. And you can see that 1 mil of the original culture was transferred into 9 mils of diluent. So diluent is the what's in the dilution test tube, the volume of, in this case, saline. It could be nutrient broth, whatever, that you add the volume transferred into the diluent. So 9 mils of diluent. And what is the formula to calculate the dilution factor from the original culture to tube number 1? What's the formula for calculating dilution factor? volume transferred over total volume. That's right. Volume transferred over total volume. So in this case, it's 1 plus 9 is the total volume, so 1 over 10. Now this could be depicted as 1 to 10, like it is in the diagram, but I don't want you to write your dilution factors that way. What's 1 divided by 10 anyway? 0.1. Did you say 1? 0.1, right? 1 divided by 10 is 0.1. What's 0.1 in scientific notation?
1.010 to the negative 2? No, what? Just 10.
It's 10 to the negative 1.
Yeah.
Yeah, 10 to the negative 1, right? And that's the way I want you to write your dilution factors. In scientific notation, you don't need to put 1 times 10 to the negative 1, just drop the 1. Right, no need to, any number times 1 is just that number, right? So 10 to the negative 1. So we would say that this one-step dilution here is creates, it's a 10 to the negative 1 dilution, which since this is the first dilution tube, then the total dilution factor in that tube is 10 to the negative 1. Now, each of these steps here is a 1 mil transfer volume into 9 mils of diluent. So every single one of them, in this case, is a 10 to the negative 1 dilution. Okay, right? Am I wrong?
Should be.
Right? They're all 1 into 9 mils of diluent. So each step is 10 to the negative 1. Okay, everybody with me so far? Okay, so what is now we have to calculate the total dilution factors of each dilution tube. So that's the TDF, the total dilution factors of each of the tubes. So to do that, you multiply all the dilution factors up to that tube, right? So for tube number 2, that's two dilutions occurred up to tube number 2, right? So to calculate the total dilution factor in dilution tube number 2, we need to multiply 10 to the minus 1 times 10 to the minus 1. So what's that number?
10 to the negative 2.
10 to the minus 2, right? Now, there are several advantages to using scientific notation, and here's one of them. When you multiply numbers like 10 to the minus 1 times 10 to the minus 1, all you have to do is add the exponents. Minus 1 plus minus 1 is minus 2. And if you want to kind of wrap your mind around that, 10 to the minus 1 is 1 over 10, right? Okay, so fine. 1 over 10 times 1 over 10 equals 1 over 100. What's 1 divided by 100? 0.01. And in scientific notation, that is 10 to the negative 2. Okay? So you don't have to do this math when you're already in scientific notation. Okay? So that means since each of these steps from one tube to the next is a 10 to the minus 1 dilution, just add, so what's 10 to the minus 2 times 10 to the minus 1 is 10 to the minus 3. So the TDF of tube 3 is 10 to the minus 3. And then 10 to the minus 3 times 10 to the minus 1 is 10 to the minus 4. So the TDF of tube 4 is 10 to the minus 4. And then 10 to the minus 4 times 10 to the minus 1 is 10 to the minus 5. So that's the TDF of the dilution tube number 5. Okay, so fine. We've now finished the serial dilution. Now what you would do when you're actually doing this is you would make a transfer into the tube. And what do you need to do to this tube before you do the next transfer? You need to mix it, and you better mix the moonshine jug up really good first, right? So you're going to mix your original culture up first, do that first transfer, mix this tube up really well, right? And then do the next transfer, mix the tube, next transfer, on and on and on. And then even when you get to the fifth and final tube, you still need to mix it because you are going to be pulling an aliquot out of it to inoculate a plate. Okay? So a lot of mixing going on there too, as well. Now once you're done with the serial dilution, then you just need to inoculate your plates and incubate them. So in this example, you can see that they pulled an aliquot from every single dilution tube and inoculated a plate for every single dilution tube. You're not going to do that. You're going to only do 4 plates, okay, from 4 dilution tubes. So there's 5 here. You're going to do 4. And let's just look at this one. From tube number 4, there was, you can see the volume used, the plated volume was 0.1 mil. And then the plate after incubation says there were 50 colonies on it. So I'm just going to write 50 CFUs. Okay? So we can use, this is the only plate that's within our 30 to 300 range, right? 389 colonies, too many. 2 colonies, too few to use, okay? But there's that one plate that's in the Goldilocks range, between 30 and 300, okay? By the way, each of these plates, the dilution factor of each of the plates is off by a factor of 10, right? So give me a theoretical count for this too numerous to count plate. How many, if you could count the colonies on that plate, how many colonies would you expect to count?
Over 1,000.
Well, it's off by a factor of 10 from the plate that's got 389 colonies. So the theoretical number of cells that went on that plate was 3,890. Right? Every plate's off by a factor of 10. That means that it's almost impossible, if everything goes correctly and you do everything right, it's almost impossible to have 2 of your plates to be in the 30 to 300 range. There's one exception, and that is if you have one plate that's 30, then you would expect another plate to have 300. So it's not impossible to have 2 plates in the 30 to 300 range. But they're all off by a factor of 10, so you would not expect that to happen. Does that make sense? Okay. So now you know, so you've done this serial dilution, you've calculated what the total dilution factor of each of the dilution tubes is. Now, you need to calculate what the plate dilution factor of this plate is. So let's do that. So the plate dilution factor, the formula for plate dilution factor, can anybody tell me? Does anybody know? Is the total dilution factor of the tube, so the TDF, times the volume plated. Okay, that's pretty easy. Got the total dilution factor of tube 4 is, as you've already calculated, 10 to the minus 4. And the volume plated is 0.1. So what's 10 to the minus 4 times 0.1? 10 to the minus 5. Remember, 0.1 in scientific notation is 10 to the minus 1. So if I erase 0.1 and I put 10 to the minus 1 in there instead, 10 to the minus 4 times 10 to the minus 1, add the exponents. Minus 4 plus minus 1 is minus 5. So the plate dilution factor for this plate right here is 10 to the minus 5. Everybody with me so far? Okay. Now that we've made all those calculations, we have one more calculation to make. That is for the OCD, right? This is what we're trying to do to begin with. And There's a long formula and a simplified version of that formula that you could use. Who can tell me what the long version of the formula is? CFU over what? Over volume plated times 1 over the total dilution factor of the dilution tube. Right? So, okay. I'm going to erase this now. Let's do this calculation. OCD equals, okay, CFUs 50 over volume plated 0.1 times 1 over the total dilution factor of the tube. So dilution tube 4, the total dilution factor is 10 to the minus 4. Okay? We can simplify that a bit. We can say it's 50 times 1 is 50. And what's 0.1 times 10 to the minus 4?
10 to
Does that sound familiar? Did we just do that?
yeah.
It's 10 to the minus 5. Okay? By the way, you just simplified this formula. CFU CFU times the plate dilution factor. Not times. What did I do wrong there?
Divide.
CFU divided by the plate dilution factor. Okay? And you can see plate dilution factor equals TDF times volume plated. TDF, volume plated, right here. CFU over PDF. And you just, right? We just did, you did this, 10 to the minus 4 times 0.1 to calculate the PDF of 10 to the minus 5. Okay? So then this becomes $50 times 10 to the 5th, doesn't it? Right? So you need to understand that 1 over 10 to the minus 5 equals 10 to the 5th. Right? So you could also think of it like this. 1 over, let's see here, draw it slightly differently. 1 over 100,000. Because 10 to the minus 5 is 1 over 100,000. Right? So 1 over 100,000 equals 100,000. Right? And 100,000 is 10 to the 5th. Everybody see that? The beauty of scientific notation. 1 over 10 to the minus 5 equals 10 to the 5th. Okay? So, that is almost the correct answer, but we've got to make one small adjustment, don't we? We've got to move the decimal from here to the left one spot to make it 5.0. So that's a plus 1. So 5 plus 1. That makes it 10 to the, that makes it 5.0 times 10 to the 6th. And then now we want to make sure we put our units back in, CFUs per mil. Okay? And that's the answer. 5 million CFUs per mil. 5 million. Okay? Does that look hard? Hector, don't convince yourself that this is hard. Okay? That's a mistake. If you convince yourself that it's hard, then it's going to be hard. But I want to stress to you how important it is for you to be able to solve these problems and solve them efficiently fairly quickly. Because they're going to pop up on several quizzes, including lecture quizzes, all this kind of math. Because this material is also covered on the lecture side of the course. That's actually where I got that picture from. This diagram is out of your textbook. Okay? So, but experience has taught me that this is one of the most challenging concepts for microbiology students. Top 3 for sure. Challenges. Okay. So I have provided you, I've tried to provide you a number of resources that will help you learn this material, because really it's just a matter of practice, practice, practice. Right? You just need to practice, need to solve some practice problems over and over and over again until you have mastered this. That's what you need to do, because there's a lot of points that you're going to miss, a lot of points if you don't do that on multiple quizzes, and including the lab and lecture finals at the end of the semester. Okay, so there are practice problems with solutions in the lab module. There's multiple video tutorials as well, and then there's an AI tutor also. So I've tried to provide you with a number of different resources. Use one of them, use both of them, use all of them. It's up to you. But use them. Practice, practice, practice. Okay? Because I make this look really easy. Not because I'm any smarter than you, only because I've just done this a lot. That's the only difference. Right? And you do some practice on your own, and you can get pretty proficient at this as well. Okay? So let's start some practice right now. Do you think you can solve a problem? A real world problem? Let's say that this is a problem on your next lab quiz. Because guess what? Your next lab quiz is going to be when?
Next Thursday.
Next Thursday. Okay, it's going to be next Thursday. And this might as well be a problem on the next lab quiz. Right there. So I want you to solve that problem. Okay? Calculate OCD. And show me your answer. I'm seeing several correct answers, but I'm also seeing some of you making some common mistakes. So I'm going to try to give you some pro tips that'll help you avoid those mistakes. First of all, let's go through this, okay? So you've got 4 dilution tubes. They're all 1 mil transfers into 9 mils of diluent. So those are all 10 to the minus 1 dilutions, right? Just like in the last problem we were looking at. So which plate should you use to calculate OCD?
C.
Plate C. It has 125 CFUs. It's the only one that's between 30 and 300. Okay? So plate C is attached to tube 3. Okay, so this is plate C, and it's got 125 CFUs. Right? So what's the total dilution factor of tube number 3? 10 to the minus 1 times 10 to the minus 1 times 10 to the minus 1 is, just add up the exponents, minus 1 plus minus 1 plus minus 1, 10 to the minus 3. Okay. And what volume is plated? 1. Not 0.1, 1 ml. Okay. So what is the plate dilution factor on this plate? Remember, the formula for plate dilution factor is the TDF of the dilution tube times the volume plated. You just calculated the TDF to be 10 to the minus 3. The volume plated is 1. So what is the plate dilution factor of this plate?
10 to the minus 3.
Stays 10 to the minus 3. 10 to the minus 3 times 1 is 10 to the minus 3. So the PDF is 10 to the minus 3. And if we use the formula for OCD of CFU over PDF, now we have everything we need. 125 over 10 to the minus 3. Right? So that becomes 125 times 10 to the third. And now here's where I've seen a few mistakes. You've got 125. You know it needs to be 1.25 times 10 to some exponent. That's not a problem. It's what the exponent is that sometimes creates confusion. So you've got 125 times 10 to the third, you need to get the decimal right there, so it's 1.25, and you're moving it 2 places from right to left, that's a +2. If you're moving it from left to right, it would be a minus however many spaces you move it. But when you're moving it from right to left, it's a plus. So +2. So 3 plus 2 is 5. And then, of course, we're going to add on our unit, CFU per mil. So the answer is 1.25 times 10 to the 5th CFUs per mil. Okay? So one of the resources that you have in the lab module is a PowerPoint file, and it has problems similar to this. And then it also, the next slide will have the solution to that problem similar to this. Okay? What I encourage you to do is don't look at the solution until you've tried to solve the problem first. If you look at the solution, you'll look at it and go, oh yeah, yep, that looks pretty easy, I could do that. But you're not learning anything. And then when you see a similar problem on the quiz, you're not going to know how to solve it. So solve the problem, or try to solve the problem first, then look at the solution. If you solve the problem incorrectly, hopefully you can see what your mistake was by looking at the solution. Hector?
You have to memorize the equation.
Yes, you need to know the equations. Yes, there's only a few. There's only a few. Good question, though. Okay, so now I want you to scroll down in your in the quick reference guide, and you're going to find that diagram right there. Okay, this is a diagram of what you are actually going to do today, starting in a few minutes. Okay, this is exactly what you're going to do today, and then below that somewhere you're going to see a data table that looks like this. Right now, what I want you to do is calculate the plate dilution factors of your plates A, B, C, and D, and then write them in. That's the very first row there of this table. DF of plate. It's asking you to write in the plate dilution factor of those 4 plates. The second and the third rows you won't be able to fill in until you evaluate the plates after they've incubated. So you won't be able to fill that information in until Tuesday. So right now what I want each group to do is calculate using this diagram right here. Hold on, let me go back to it. Using this diagram right here, I want you to calculate the plate dilution factors of these plates. Attention to detail. Pay attention to your volumes transferred. Pay attention to your diluent volumes. And pay attention to your volumes plated. They're not always going to be the same. As long as you're plugging them into the correct formulas, you're going to get the correct answers. Okay? So go ahead, calculate those PDFs, and then show me your calculations when you're done. Okay? Okay, let's look at this. All right. I've drawn the diagram up here, as messy as it is. Same diagram. I mean, same as what I've showing up here. In order to calculate the plate dilution factors of plates A, B, C, and D, you need to first calculate the total dilution factors of the dilution tubes, right? So using the formula for the dilution factor, volume transferred over total volume, got a 0.1 mil transfer here into 9.9, so that is, that's 0.1 over 99. That's 1 over 100. That's 10 to the minus 2. So since that's the first tube, the TDF of tube number 1 is 10 to the minus 2. There's no plate from that tube. The next step here is again the same as the first step. It's 0.1 into 9.9. So that's another 10 to the minus 2 step. But to calculate the total dilution factor of tube number 2, it's 10 to the minus 2 times 10 to the minus 2, so it's
10 to the minus 4.
10 to the minus 4. So the total dilution factor in tube number 2 is 10 to the minus 4. And then we go to 1 into 9. So that's a 10 to the, what? 10 to the minus 1. And so the rest of these steps are 1 into 9, so they're all 10 to the minus 1 steps. So 10 to the minus 4 times 10 to the minus 1 is?
Is
10 to the
5.
minus 5, so the TDF of tube 3 is 10 to the minus 5. 10 to the minus 5 times 10 to the minus 1 is?
10 to the minus 6.
10 to the minus 6. So the TDF of tube 4 is 10 to the minus 6. And then 10 to the minus 6 times 10 to the minus 1 is?
10 to the minus 7.
10 to the minus 7. So the TDF of tube 5 is 10 to the minus 7. I'm getting ahead of myself a little bit there. Okay? So now we've calculated the TDF, the total dilution factor of each of the dilution tubes. And now we need to calculate the PDF, the plate dilution factors of each of the plates. The formula for PDF is total dilution factor of the tube times the volume plated. Okay, let's start at plate A. TDF is 10 to the minus 4 times 0.1, so the PDF is, what's 10 to the minus 4 times 0.1? 10 to the minus 5. 0.1 is the same as 10 to the minus 1. 10 to the minus 5 times 10 to the minus, 10 to the minus 4 times 10 to the minus 1 is 10 to the minus 5. So the PDF here is 10 to the minus 5. For plate B, that's a weird looking 5. Let me fix that. 10 to the minus 5 times 0.1.
10 to the minus 6.
So the PDF of plate B is 10 to the minus 6. For plate C, 10 to the minus 6 times 0.1.
10 to the minus 7.
So the PDF of plate C is 10 to the minus 7. And for plate D, 10 to the minus 7 times 0.1 is 10 to the minus 8. So the PDF of plate D is 10 to the minus 8. Okay? When you're labeling your 4 plates today, you're going to put all the usual stuff on your labels, date, group number, etc. You are also going to put the letter A, B, C, or D and the PDF on each plate. Make sure that the PDF is also on each plate. It's part of your labels. Okay? Any questions about how we got that? No questions?